The discovery of novel antibiotics and other therapeutic drugs is a major driving force behind advancements in medicine and global health. New methods of drug design, biosynthesis, and bioactive metabolite reprogramming have revolutionized the way we treat various diseases such as cancer, as well as bacterial and fungal infections. My name is Jia Tanwani, and I'm one of the co-directors of Queen's QGEM. To supplement our project this year on novel drug discovery and optimization, as well as related projects engaged by other QGEM teams, we've decided to film a 3-part interview series. This interview series aims to address overarching questions related to biosynthetic production, aspects of drug design, metabolite reprogramming, and much, much more from a variety of unique perspectives. We welcome you to join us through each of the 3 segments as we explore perspectives from research, the industry, and a biosynthetic startup. To start off, I'd like to introduce Dr. Michael Adams. Dr. Adams is a professor and the head of the Department of Biomedical and Molecular Sciences here at Queen's University and has quite an extensive background in pharmacology. Today we'll be interviewing him to learn about the drug design process in depth, all the way from the initial planning stages to the clinical trial stages. Welcome, Dr. Adams.
Thanks for having me.
So tell us a little bit about your educational background. So what you did after high school and what sort of led you there?
After high school, I decided to go into basically a biology program somewhere. I didn't quite know where it was going to take me, so I was accepted to both, actually Queen's, to Guelph, and Western. And since I lived in Montreal, I went far away from home, so I went to Western. Looking back, I probably should have come to Queen's, but I chose Western simply from the point of view of a 17-year-old trying to go somewhere else.
Okay.
Went there and I realized that as I was looking for 2nd and 3rd and 4th year courses that I was really interested in very much applied kind of things. And when I mean application, I want translational, I like clinical.
Okay.
I wasn't so interested in just pathway analysis. I wanted to do something that really had an application in human health. So I started really thinking around what I did. In 3rd year I took a pharmacology course, really loved it. And in 4th year I applied for a very limited enrollment physiology, pharmacology, 4th year program, got into it. There was only 16 of us.
Wow.
Spent, oh Lord, it was 25 hours a week together in labs doing stuff. It was a lot. And as a result, I learned a lot. And then I went into grad school. And it was my driving force. So grad school, I did a master's, thought I might leave Western to go somewhere else, and then thought I have a really good project, stayed at Western, finished my PhD quickly, and then postdoced in Australia. So it was a fast track very quickly because I wanted to do something translational. I knew that right from the get-go. Yeah.
Wow, good for you. So what was the focus of your PhD?
My PhD was actually on alcohol.
Okay.
I actually started in a project where I found some very interesting things about binge alcohol intoxication. And no one really knew much about what the negative attributes of binge alcohol intoxication, so I discovered very early in my master's that it produced a hyperactivation of the sympathetic nervous system and that the release of catecholamines into the circulation actually charged around the body and did some crazy things. And one of the most remarkable things that we found was the enlargement of the heart, hypertrophy of the heart, both left and right ventricles, and actually atria, in a very inappropriate way, in a very maladaptive way. In fact, it created a process where even parts of the heart would be infarcted, like acute myocardial infarctions. And it did so in less than 4 days. So we became aware that, wow, people were not sure of this, but our data in animal studies showed that if somebody binges excessively and they are prone to excessive catecholamine release, they could have serious risks in terms of cardiac. So then we examined what receptors are involved, and I ended up getting about, I don't know, out of my PhD and master's, I got about 12 papers,
Wow.
which got me a postdoctoral fellowship, and that's when I jump-started. And I left that. I mean, alcohol was a cool place to start. The cardiac hypertrophy took me into another realm of cardiovascular growth responses,
All right.
which is what I did in my postdoc.
Okay. So I've previously heard that you were part of bringing a drug to market. So could you talk more about what that drug was, what its and what its intended therapeutic effects were?
Yeah, so this is a long time ago now. And I've been involved in both bringing a drug to market as well as a startup company. And before we had the startup company, we had an idea that some of the studies we were doing actually could have a human application, and they were in the world of sexual medicine. So I had worked, as I was just telling you, I was working in cardiovascular work, cardiac hypertrophy, vascular hypertrophy, hypertension even, and a urologist walked through my door. And he's a surgeon, right? But a surgeon who worked in the area of male sexual health.
Okay.
And we started doing research together, and after about 3 years of work together, we realized that there was an opportunity to take the drug we were using in animal studies to take it to the market. And it was pretty exciting. So we thought, well, what do we need to accomplish? And so we basically invented a drug at Kingston General Hospital and in our labs, which was a sublingual formulation of a non-selective dopaminergic agonist that, because of its particular attributes, arrived into the brain, hit nuclei in the hypothalamus and triggered sexual responses, the beginnings of sexual responses. When added to sexual responses that were natural, generated erectile responses. So we spent some time trying to perfect what kind of formulation it was, had a really good run of experiments in males, obviously, and then hooked in with a company out of Chicago who advanced it another layer to manufacturing stage, preliminary manufacturing stage, and again showed very, very good success with very minimal adverse effect profile. And then we moved on, and we ended up with a deal with Takeda Abbott Pharmaceuticals, who threw all their resources at it. They were called TAP, Takeda Abbott Pharma. They threw a ton of resources at it, hundreds of millions of dollars, and over a number of years, we pushed it to market by literally, you take ours were phase 2-3 preliminary trials. They weren't sufficient for registration. You have to use what's called a phase 3 pivotal trial, which is really a large scale, a lot of patients, a lot of diverse patients with really defined outcomes. The only issue is that you got to get the formulation right.
Okay.
So the formulation we had in the first round and the second round worked perfectly, but they were not scaled to phase 3 pivotal. They were not scaled to marketing, so they took it to a bigger firm to get that done, and the formulation wasn't quite the same.
All right.
We recognized that. We told the company and said, there's something not quite right here. No, it's the same. All the dissolution characteristics are identical, blah, blah, blah. It ended up not being the same. Meanwhile, $300 million later, you've got a drug that's had a whole bunch of testing on it, and 2 and 3 milligram doses work fine in terms of no adverse event profile, but their efficacy was lower. When you moved up to 4 and 5 milligrams, the adverse event profile went up, which was not acceptable in what's thought of at the time as a lifestyle drug, right? Before Viagra. This was before Viagra. It made it to the market in Europe, in Asia, in Africa, in the Middle East, in Australia, in the Caribbean, South America.
Everywhere except for North America.
Everywhere except North America. But exactly at the time it was coming on the market, right, or when we were trying to get to the market, came Viagra out of the blue. You can imagine the term we like to use was gobsmacked. We were at a meeting in Orlando when the results of Pfizer brought to the table on their drug coming out. We were just going, oh no, because their drug was oral, very minimal side effects, adverse events, but had very good profile, and obviously it's done very well. It doesn't affect everybody.
Yeah.
So our drug stayed on the market for about 6 or 7 years, never made a ton of money, but it clearly got people thinking about central nervous system acting drugs to the extent that it was the one example that brought the companies to bear in female sexual dysfunction, and there's now a drug approved in the last 6 months for female sexual dysfunction, which is a centrally acting drug. Obviously, we had to do things like patent it starting in 1992, and advance patents on, and patenting is a crazy world. Patent's a fun and crazy world. It's a completely different language. You have to learn what a claim is. You have to learn how to defend your claim. You have to learn how to have appropriate discussion with an examiner, disagree with them, get them to go onto your side. Similar to reviewers of papers, but very different. Of course, all those attributes helped a lot when in 1996, 97, we started the company. And that was, again, the next layer of, if you want, the commercialization of intellectual property. And at this time I had a graduate student who was working with us on it, and he was the perfect guy to jump into that, and he finished his PhD. His name was on 3 of the patents, and he decided to not go off on his own. He wanted to be part of the company. So we said,
Great.
So he jumped into the company with us. And we got funded by Canadian Medical Discoveries Fund for $5 million. And we were an operating company from about 96 onward. And then we sold our company actually about 4 years later to a company out of San Francisco. So,
Okay. And how come the decision to sell?
In Canada at the time, there was resistance to fully fund a Canadian company to do the full tilt with full-blown trials, and what was needed was the deeper pockets of an American company who could access, quite frankly, deeper pocketed financial institutions in the US. So we stayed involved with them and saw them as an opportunistic company, thought that they had the right flavor. In the end, some of the patents came back to us because they couldn't close the deal.
Okay.
And some of them are still ongoing, but it's a different world. I mean, I was trained as a scientist, right? And as a scientist and educator, and to then step into the business world, you got to pick up a whole new lingo and a whole new strategy.
Interesting.
Yeah.
So you find that scale up is the biggest challenge in taking an idea and making it real.
Yep.
As well as patenting.
Patenting, after a while, I don't fail at patenting now.
Okay.
I know what's intellectual property. I know what's patentable. When you do the legwork, you always can find the niche that gives you a patent. A usable patent plus usable idea plus ability to implement and ability to scale up.
Okay.
Those are way tougher at the back end.
All right.
I've had 16 independent inventions patented,
Wow.
which gives me worldwide hundreds of patents. That's not hard anymore.
Okay.
That's neat. I've got them. I can show you the stack of first pages of my patents. They're just littered, invented by kind of thing. But that's okay. But getting a drug to market is a bigger deal. Getting a company fully formed, able to implement, to scale up, and to get it through all the morass of the marketplace is the far tougher.
Now, do you think scale up was harder in your time, so in the 90s, or do you think it's harder nowadays when there's more competition?
I think it's tougher now.
Okay.
It's tough then, it's tougher now.
Okay.
I mean, some people make it through. I'm still working with a company right now. One of my friends, he started up his company exactly the same time as we did.
Okay.
This is Dr. Martin Pekovich, okay? So, and they had cytochroma. Cytochroma started exactly the same time as we did with Vaxxas. Our company was called Vaxxas. And he stayed with it, trucked along, took a leave of absence for several years, and then cytochroma was bought by OPCO. Now success is happening because OPCO, deeper pockets in the US, run out of Florida, and they just had a drug approved.
Wonderful.
So it's, there's staying with it and fighting tooth and nail to go with that. So you have to be very, very patient.
Okay.
And they started at the same time, so we're 2016. He started in 1996.
Wow.
So long haul.
So you have to be persistent when
Very
it comes
persistent,
to it.
yeah.
And there's obviously, like you said, you have training in science, but you have to translate that into the entrepreneur world,
Yeah.
where things are run very differently.
Very differently. You can imagine going into a room where, here's one story for you. So when we were Vaxxas, we had discovered something very, very interesting in Jim Banting's thesis. Okay, so Jim was my PhD student, and one of these findings is that we knew how to test this class of drugs called endothelin antagonists,
Okay.
I think, better than anybody else. And what Jim had discovered was an attribute of the drugs that we could examine quantitatively, and so we had the patent, we had a worldwide patent for use of these drugs in sexual dysfunction.
Okay.
These companies were interested. They were interested in any target. So they agreed, it was about 8 companies gave us close to 40 compounds. Their patented specialty compounds, they gave them to us to test.
Gotcha.
So what did we find out? We found out that more than 95% of them were useless. That they would not do the job, and we told them so. Now hang on. So here's these scientists from Queens who are saying to these scientists from companies who have just spent $100 million on the development of the molecule, that their molecule wasn't very good. So how did that go over? Not well sometimes. They were mortified, they were defensive, and they challenged us to the nail. We're going like, you asked for our advice, we were talking about a partnership, but we're telling you that we don't want a partnership with you on this because we don't think your drug has the properties to make it effective in this particular condition. in the end, when you examine our success, look at the number, if you typed in today, how many endothelin antagonists are on the market? 1. They did not make it. They did not have the efficacy to do it without having adverse event profiles. They designed drugs without the right testing modules. They did not do the right things, and we could tell at the time that they made a mistake. So that we told them that in 1998.
Wow.
And no, endothelin was only discovered in 1988. That was only 10 years after the molecule was discovered to have actions in the body. And then for us to tell them 10 years later that they were getting it wrong, we're talking $10 billion was spent worldwide, and we tested all the major companies and told them their drugs were terrible.
Wow.
So we realized, Jim and I said that, we were the world experts on testing these molecules. They should have given them all to us. We knew how to do it, and that's what we should have done. That was our niche that we didn't realize at the time.
Ah.
So we could have probably saved them a lot of time and energy. We knew which molecules were really good. One of them was a peptide with a duration of action of about 2 minutes.
Okay.
Useless though, 2-minute molecule. So you put it into your body and then it's gone.
Yeah.
So the company couldn't work with that. They had the peptide, it was a peptide. Well, it works, but as soon as you stop infusing it, it stops working.